Good evening, everyone. So, we'll start in 5 minutes so that more people can join in. Okay. Just, I guess, one more minute and we'll start. Okay. I think we can start our today's session. So, if you remember what we were discussing in the last class, we were going through the affinity maturation of B cells. We were seeing how the B cells see they are selected, and then somatic hypermutation, how they are allowed to proliferate, how they are developed with the help of helper T cells. Okay, so we were seeing how basically the immune system is working in the lymphoid organ, and especially in the lymphoid organ, what happens exactly at the germinal center. So if you remember, this was the germinal center. Now you know in the dark zone and in the light zone what kind of cells are predominantly present. Okay, so now continuing our discussion. Yeah, so this is basically the features of antibody responses to T dependent and T independent antigens. So T dependent antigens, that the proteins predominantly, and T independent antigens, it can be anything, includes antibody response with different characteristics, largely reflecting the influence of helper T cells in T dependent responses to protein antigens, and the absence of T cell help in T dependent responses. Okay, so if you see in the thymus dependent, the chemical nature of the antigen is predominantly protein. Okay, and the thymus independent process, the type of antigen can be polymeric antigen, polysaccharide, glycolipids, nucleic acid, it can be anything. Now when we talk about the features of antibody and the responses which they are eliciting, so in the thymus dependent part, the heavy chain switching or the isotype switching occurs. So earlier it will be IgM, and then if it has seen the antigen, then it will go to IgG, IgE, IgA, depending upon what sort of interaction is needed at that particular time. Now when we talk about in thymus independent antigen, so there is a very low level of class switching, or say not much class switching is there. Okay, so predominantly in that case the majority of the antibody will be of type IgM, and very small quantities of IgG will also be there. When we talk about affinity maturation, so obviously if it is happening with the help of thymus, or say with the help of T helper cell, the affinity maturation will definitely take place. Okay, now if we talk about thymus independent antigen affinity or antibody affinity maturation, as it has not taken any help from the T helper cell, there will be very little or very low amount of affinity maturation will be present. Now when we talk about plasma cells, which are basically the antibody secreting cells, they'll be long lived, so that the high affinity, highly specific antibodies can be secreted for a long duration of time. But when we talk about thymus independent maturation, they are there just for the immediate response. Okay, so for the immediate response they'll be very short lived. If we talk about secondary responses or memory B cells, okay, so in both the cases, if you remember this particular graph, so the plasma cells will be there, but there is no development of memory cells, and even if the memory cells are there, they are very low in number. So there is a very not so clear distinction between whether the memory cells will be there or not. But if we talk about thymus dependent and thymus independent maturation, then for sure we can say that in the thymus dependent maturation, the memory B cells will be there, hence it will be more specific, more pronounced, and the serum antibody level will be high. Okay, when we talk about thymus independent antigen B cell maturation, then what happens is that the memory cells are almost negligible. If we compare it with thymus dependent one, they'll be clearly no. Okay, but if we say if there is little bit memory associated, then negligible amount of memory B cells will be present. Okay, but if we are comparing both of these, like the thymus dependent and thymus independent, we have a clear cut answer that there will be no memory cells associated with the thymus independent antigen B cell maturation. Okay, and as this might seem familiar as well as a little bit difficult, but we have already covered this. Even in the class lectures, this topic has been covered. So at what stage what happens, or basically the sequence of events and characteristics of the stages in the B cell maturation. So B cell maturation takes place in bone marrow. So the stages, so what will be the stages? The stages will be, first, multipotent cell or lymphoid stem cell will convert into pro B cell, then pro B cell will convert into pre B cell, then pre B cell will convert into immature B cell, then immature B cell will convert into naive B cell, then naive B cell will convert into mature B cell. This mature B cell will further go to the lymphoid organ, and then there it will see the antigen and it will get activated, and all the process of isotype switching, somatic hypermutation, all those things will take place. Okay, so this is the brief overview of the processes which are happening at every single stage of B cell maturation. So first, when lymphoid stem cell is getting converted to pro B cell, so what exactly is happening? The D and J joining is happening. Okay, and at this time the immunoglobulin alpha or beta chains will be displayed. When pro B cell is converted to pre B cell, at this time the V segment will be joined with the recently formed DJ segment. Okay, when another surrogate light chain for the pre-BCR will be there, which means there is no light chain for BCR at this stage till now. Okay, after this, if this is good enough and the cell is okay, it doesn't needs to undergo any apoptosis, then another V segment will join to the J segment, because we need more variety. Okay, we need IgM type of antibodies which will be there first. Okay, then random combinations will happen, and then immature B cell will convert into naive B cell. So this naive B cell has B cell receptor, has co-receptors. Okay, and then in the mature B cell you have all these receptors which are necessary for a B cell to be recognized as a B cell. And these are the things which are there. You don't need to worry about each and every row of this. This is just the summary. You have already been familiarized with each of them in a separate lectures. Okay, so this is just like a summary to what happens and what processes or what events are occurring when a B cell is maturing. Okay, again, these are just some overview of signaling. So why we are concerned about the B cell receptor, why we are concerned about how the segmentation or mutation or isotype switching is happening. So every single thing is led by the signaling. So again, this just to make sure that you understand the concept of signaling, that they are not only for the immunology perspective, they are general signaling pathways. Okay, and each pathway can be stimulated by different ligands or different receptors. Okay, so for example, in this you can see the major pathways which are there is G protein pathway, PKC pathway, and calcium mediated pathways. So these three pathways we have already discussed here. See, GTP or G protein pathway, calcium dependent pathway, and DAG. Okay, so these are the similar things, but it's just they are represented in a different way. So there it was given just the transcription factors. Here the processes which are involved or which occur due to the activation of those transcription factor is enlisted. So it leads to the changes in the pattern of gene expression, that means via the transcription factor. Functional changes in the cells. So when the gene expression pattern changes of a cell, then definitely the functional change in the cell will take place. Then the differentiation, now the cell actually needs to adapt to the changes which it just got communicated with. Okay, and the cell will get activated. So this is just a side information so that you can relate to what exactly those transcription factors are doing. So those transcription factors were released because some enzymes were activated. Why some enzymes were activated? Because some proteins were there in the cytosol. And why those proteins were there in the cytosol? Because a ligand has just came and bound to the surface receptor. If we talk about B cell, then it will be antigen. If we talk about T cell, then it can be a MHC receptor, okay, from APCs. Now a small summary about whatever we have covered in the previous two lectures. Okay, so the humoral immunity is mediated by antibodies that bind to extracellular microbes and their toxins, which are neutralized or targeted for destruction by phagocytes and the complement system. So we're gonna discuss about the complement system today. Humoral immune responses are initiated by recognition of the antigens by specific membrane immunoglobulin, antigen receptors of the naive B cell, the binding of multivalent antigen cross-links B cell antigen receptors of specific B cells, and biochemical signals are delivered to the inside of the B cell by Ig associated signaling proteins. So one such protein is ITAM. Remember, we discussed in the last class. These signals include B cell clonal expansion and IgM secretion. Okay. In humoral immune responses to a protein antigen called T-dependent responses, the binding of protein antigen to specific Ig receptors or naive B cells in lymphoid follicles result in the generation of signals that prepare the B cell for interaction with the activated helper T cell that expresses CD40 ligand, okay, and secrete cytokines. The B cells internalize and process that antigen and present class 2 major histocompatibility complex displayed peptides to activated helper T cells specific for the displayed peptide MHC complex. These helper T cells contribute to early B cell activation and extrafollicular sites The early T-dependent humoral response occurs in extrafollicular foci and generates low level of class-switched antibodies that are produced by short-lived plasma cells. Activated B cells induce the further activation of T cells and their differentiation into T follicular helper cells. The B cells, together with the T follicular helper cells, migrate into follicles and form germinal centers. The full T-dependent humoral response develops in germinal centers and leads to extensive class switching and affinity maturation. The generation of long-lived plasma cells that secrete antibodies for many years and development of long-lived memory B cells which rapidly respond to re-encounter with antigen by proliferation and secretion of high affinity antibodies. So you might think that these are random text or something, but this is the overall summary of what we have discussed from here. The phases of humoral immune responses. So here we are talking about the activation of B lymphocytes, we are talking about the secretion of antibodies, we are talking about the B cells maturing and differentiating, then we are talking about different types of IgG, then we are talking about how they are basically maturing. So this we have discussed in two separate classes, hence this summary might seem a little bit aloof, but it is the continuation of whatever we have discussed. Okay. Then affinity maturation is the process by which the affinity of antibodies for protein antigens increases with prolonged or repeated exposure to the antigens. The process is initiated by signals from the T follicular helper cells, resulting in migration of the B cell into follicles and the formation of germinal centers. Here, the B cells proliferate rapidly and their IgV genes undergo extensive somatic mutation. The antigen may be displayed by FDCs in the germinal center. FDCs are follicular dendritic cells. Okay. B cells with mutated V regions have recognized the antigen with high affinity are selected to survive, giving rise to affinity maturation of the antibody response. Polysaccharides, lipids, and other non-protein antigens are called T-independent antigens because they induce antibody responses without the help of T cells. Most T-independent antigens contains multiple identical epitopes that are able to cross-link many Ig receptors on a B cell, providing signals that stimulate B cell responses even in the absence of helper T cell activation. Antibody responses to T-independent antigens show less heavy chain class switching and affinity maturation than is typical for responses to T-dependent protein antigens. Secreted antibodies from immune complexes with residual antigen and shut off B cell activation by engaging in inhibitory Fc receptor on B cells. So this is the overview of cellular events within the germinal center. Okay, so these are just different way of representing it. So whatever you have seen here, and I am extremely sorry that I am going to and fro little bit, but I want you to understand that everything is connected. So this was a representation of the germinal center and the events which are taking place in the germinal center, right? This is also the same thing. It's just it is represented in a different way. So if we see here, We'll just read the legend for this first. Okay, so antigen stimulated B cells migrate into germinal center. So here the B cell has seen the antigen, okay, where they reduce expression of surface Ig and undergo rapid cell division and mutation to rearranged immunoglobulin V region or variable region gene within the dark zone. Subsequently, division stops and the B cell migrate to the light zone and increase their expression of surface immunoglobulin. At this stage, they are called centrocytes. So see, here is a little bit of different information of the same process. Within the light zone, centrocytes must interact with follicular dendritic cells and T helper cells to survive. Follicular dendritic cells bind antigen body complexes along their long extensions, and centrocytes must compete with each other to bind antigen. B cells bearing high affinity membrane immunoglobulin or antibodies shown here in the blue, these ones in this case. So we had blue, gray, right, these antibodies present here. Those that fail this antigen mediated selection die by apoptosis. B cells that pass antigen selection and receive a second survival signal from the Th cell differentiate into either memory B cells or antibody secreting plasma cells. The encounter with the Th cell may also induce class switching. A major outcome of the germinal center is to generate higher affinity B cells from B cells of lower affinity. So same information in different way. Now what is happening? The B cells who has seen the antigen will go into the germinal center. This is the dark zone. Okay, this is the light zone. So in the dark zone, the somatic mutation in the cells will happen, and due to somatic mutation, a lot of isotype switching or class switching will take place. Okay, so basically somatic mutation will lead to the formation of different antibodies on the surface. Few of the antibodies will be high affinity, few of the antibodies will be of low affinity. How we are saying that this is high affinity, low affinity? It will depend upon how well it is binding with the antigen showed by the follicular dendritic cells. If it can bind very strongly with it on them after competition, then they'll be allowed to be selected, and the secondary selection will occur when the same cell makes an interaction with the T helper cell. Once the T helper cell helps the B cell or selected B cell, then they'll convert into plasma cells, and plasma cells are the one which are secreting the antibodies which are just selected at this stage. Okay, and they'll also convert into memory B cells. Okay, so these are the same events which are happening. Little bit extra information is there. So like if I say that you have to write about germinal center or what is germinal center, so the first thing which you start with, that they are in the lymphoid organs, they have dark zone, light zone, major events which occur in dark zone, major events which occur in light zone. So you have good enough amount of knowledge and information to basically write something about the germinal center. Okay, that is why I'm trying to give you different perspectives of the same thing. The course is not hefty, it's just my personal view is, if you see same thing from two different point of view, it gives you a little bit more understanding of the concept. Okay, now if you talk about the effects of antibodies, so all this process happened just for the secretion of antibody, but what exactly this antibody is doing, right? So there must be important functions which are supported by the antibody, right? So the primary function is, okay, it will fight the infection, but how it is fighting the infection? Because remember, they don't have eyes, right? Since the beginning of the class, as I am saying, the cells don't see, they don't have eyes, but still they communicate via ligands, they communicate via receptors. So if now the antibody is made after so much of struggle, so much of selection, everything is done, and now we have these antibodies. Okay, so what will happen when that antibody is there? So there are few ways through which antibody acts. So antibodies are produced, you already know this, by the activation of B lymphocytes by antigens and other signals. Antibodies of different heavy chain classes or isotypes perform different function. Okay, so you should question this. If IgM, which is such a like bulky antibody, if you remember this figure, yeah, you can see this is IgM, and this is the one which is there since the beginning, like this is the first antibody. Okay, so why do we need IgG, IgE? They are very different, and this seems very small in front of IgM as well, right? So the major reason why we need multiple type of antibodies, because every single antibody works differently. They have different function. Okay, so if we take the example here, I hope you guys can see this. Yeah, if you can see here, so this is the B cell which has just seen the antigen. Okay, antibodies are secreted. So what are the pathways an antibody can take? It can neutralization of the microbe and toxins. What do you mean by neutralization? Neutralization is basically all the antibodies will bind to this microbe, and this microbe is not at all free to do anything else. Okay, hence the antigen is neutralized. Second is opsonization and phagocytosis of microbes. Okay, we'll see this in a little bit more detail today. Then antibody dependent cellular toxicity, you know this very well. It will directly kill the infected cell. Okay, then lysis of the microbe itself. We are not, the microbe is there freely outside the cell, and the antibodies will directly go and lyse it with the help of phagocytic cells. Okay, then phagocytosis of microbes opsonize with complement fragments. Other pathway is complement activation. And then one and other thing is inflammation. Okay, so now major effectors of different types of antibody. So if we talk about IgG, so you remember IgG is the first antibody in the secondary response, right? So IgG, it has what functions it has? So it does neutralization of microbes and toxins, opsonization of antigens for phagocytosis by macrophages and neutrophils, activation of the classical pathway of complement, which we will see today, antibody dependent cellular cytotoxicity mediated by NK cells, neonatal immunity, that is transfer of maternal antibody across placenta and gut, feedback inhibition of B cell activation. If we talk about IgM, then activation of the classical pathway of complement system. So you can see here IgG has so many functions which we just saw here, while IgM, the predominant function of IgM is the activation of complement system. Okay, if we talk about IgA, so the mucosa, it is said to have or provide the mucosal immunity. So the secretion of IgA into lumens of gastrointestinal and respiratory tracts, neutralization of microbes and toxins. IgE, it activates the mast cell. So they secretes the this anti-allergic factors. Okay, and then defense against helminthes. So despite these are the functions which are done by the antibody, but not every single type of antibody is doing all the functions. Okay, so these are the predominant effector functions of these particular major type of antibodies. So if you talk about IgG1 or IgG4, they'll have these functions only. It's just their amount of specificity, or say to the extent they are doing this function, will vary, but they'll do these functions only. Okay, so if we say the kiddo, a newborn kiddo, which antibody it will have? It will have say. This is a question for you guys. Okay, whether it will be IgG or IgA. Okay, so the answer is on this slide itself. Now if we talk about the neutralization of microbes and toxins by antibodies, so this is the neutralization. Okay, one function of antibody, not all the functions of antibody. So for neutralization, in the A panel without antibody and in the B panel with the help of antibody. So the antibodies at the epithelial surface, such as in the gastrointestinal and respiratory tract, block the entry of ingested and inhaled microbe respectively. I think it is a little bit self-explanatory, but yeah, let me just go through it. So antibodies prevent the binding of the microbes to cells, thereby blocking the ability of the microbe to infect the host cell. Okay, and then the C panel shows that antibody block the binding of toxin to cells, thereby inhibiting the entry. So don't complicate it, let's just see the first panel. So this is the A panel. If there is no antibody and this is our epithelial cell, okay, and this is the barrier, the microbe can enter very well inside that, right? But now imagine we have antibodies, and specifically which type of antibody? So the neutralization is done by IgG as well as IgA. Okay, so it can be either IgG or IgA type of antibody. So once it bounds to the antigen or the microbe cannot enter, so it neutralized the infection. Now imagine if the antigen is there and it has bound to the receptor of our cell, so it will infect our cell because it has already crossed the first barrier, so even the second barrier will be crossed. Now imagine if the antibody is collected here, then it doesn't have any receptor to bind to our cells or our tissue cells. Again, it saved the infection. Okay, if we talk about toxins, the same thing happens with the toxins itself. Okay, so once the antigen is there and if the antibody comes and bind to it and it neutralizes it, it cannot further infect our system. This is what we mean by neutralization of the microbe or the antigen, which is a major function or one of the major function by the antibodies. And even in the antibody, IgG and IgA type of antibodies. Okay. Second function is antibody mediated opsonization and phagocytosis of microbes. So opsonization and phagocytosis, again it is a function of antibody. Okay, so what exactly is opsonization? So opsonization means so in layman language, what we can say is that it is making thing more palatable or more tasty. Okay, so imagine this is a microbe, okay, an IgG antibody is bound to it and there is a phagocytic cell. So the phagocytic cell can bind to these antibodies which are bound to the microbe, which was freely moving here and there. Once it binds to this complex, this phagocytic cell becomes more receptive for engulfing this microbe, which earlier it was not able to see. So basically what antibodies are doing, antibodies are making the phagocytosis of this microbe more effective or more palatable for the phagocytic cell. So the antibodies of certain IgG subclass, specifically IgG1 and IgG3, bind to the microbe and are then recognized by the Fc receptor on phagocytic cells. Signals from the Fc receptor promote the phagocytosis of the opsonized microbes and activate the phagocytes to destroy these microbes. Okay. Now till here we saw what are B cells, what are T cells, how B cell is matured, where B cell is matured, and what is the purpose of B cell maturation is basically the secretion of antibody. What are the different types of antibodies? What are their major functions? This is what we have discussed. So as you can see here, there is another important function which is called complement activation, which we are listening or hearing since the first class, that there is innate immunity, active immunity, humoral immunity, cell mediated immunity. A major type of humoral immunity is also complement activation. Okay, so now we are digging into complement system. So the complement system I hope you guys are still in the class. So the complement system is a network of circulating and membrane bound proteins crucial for host defense against microbes and for antibody mediated tissue injury. Its name comes from the fact that it complements the action of antibodies in destroying cells. It can be activated in two ways, majorly like two ways, but there are in total three ways. We'll discuss it. So in innate immunity, directly by microbes without antibodies. Okay, so there are two pathways or two complement system in which there is no need of any antibody. Okay, in adaptive immunity, so by antibodies bound to microbial surface. So there is one pathway which is called the classical pathway, which initiates the complement system, where it recognizes this antigen antibody complex. Okay, so the activation proceeds through a cascade of proteolytic cleavages, producing effector molecules that eliminate microbes by mechanisms such as cell lysis and opsonization. Like other enzymatic cascades, this process allows amplification, where small initial signals generate a large downstream responses. Importantly, activated complement proteins bind covalently to the microbial or target cell surface, localizing the attack on the right place. To prevent harm to self, host cells have regulatory proteins that inhibit activation and deposition of complement components, thereby avoiding damage to healthy tissue. This might seem a little bit foreign, but what we'll do today is we'll first see the visual aid for complement system. You guys will get a little bit of idea what exactly complement system is. Then we'll go through the theory of it, like every and each step, and then we'll see the summarizing figures. Okay, so let me start with this because it's one of the important important area in the immunology, the whole complement system. So it's better to take a little bit of more time in understanding.
From villainous to sharks, from
The complement system is an important part of the innate immune response and plays a major role in the killing and clearance of invading pathogens, including bacteria, viruses, fungi, and parasites. The system is composed of over 30 proteins, most of which are found in serum, while the remaining proteins are membrane-bound proteins and receptors. On activation, complement proteins that participate in the activation of either the classical or alternative pathways of complement interact in a highly specific enzymatic cascade and generate proteolytic fragments that mediate the numerous biological functions of the complement system. In this video, we will focus on the activation and biological functions of complement. Activation of the classical pathway. The classical pathway is typically activated by antigen-antibody complexes. Although there are many other activators of this pathway, only two isotypes of antibody, IgM and, as shown here, IgG, will activate the classical pathway. While one pentameric IgM antibody will activate the classical pathway, a minimum of two IgG antibodies in close proximity are required. In either case, antibody must be bound to antigen in order to activate the classical pathway. The classical pathway is activated when C1, the first protein in the pathway, binds to the Fc portion of the antibody. C1 is a large macromolecule composed of a C1q molecule and two molecules each of C1r and C1s. It's the globular head portion of the C1q that binds to the Fc binding site on the antibody. This binding causes a conformational change in C1q and subsequent autocatalytic conversion of C1r to an active serine protease. Activated C1r cleaves C1s to an active serine protease. Activation of C1 by binding antibody is also termed complement fixation. The next step in the activation of the classical pathway is the cleavage of C4 by the activated C1 molecule. C4 is cleaved by the activated C1s molecule in C1 into two proteolytic fragments, a small peptide called C4a and a larger fragment, C4b. C4b attaches covalently to the antigen-antibody complex and has a binding site for C2, the next protein cleaved in the classical pathway. C2 is also cleaved into two fragments, and the resulting C2a fragment binds to C4b. This bimolecular complex of C4b2a is a C3 convertase of the classical pathway and serves to cleave C3 through the enzymatic activity in the C2a portion of the complex. Classical pathway C3 convertase. The C3 molecule is cleaved by the C3 convertase into C3a and C3b. The C3a fragment has potent biological activities, which will be discussed shortly. The C3b fragment can covalently attach to the C3 convertase, resulting in a trimolecular complex consisting of C4b, 2a, 3b. This multimolecular complex is called the C5 convertase of the classical pathway and is specific for cleaving C5. Classical pathway C5 convertase. The C5 molecule is cleaved by the C5 convertase into C5a and C5b, both of which mediate important host defense functions of the complement system. Both C3a and C5a are potent chemoattractants. C3a attracts mast cells to sites of complement activation, and binding of C3a and C5a to these cells, as well as basophils, induces degranulation and release of histamine and other vasoactive amines. C5a is a chemoattractant for macrophages and neutrophils, and binding of C5a to these cells primes them for mediating their host defense functions. The terminal complement pathway, or formation of the membrane attack complex. One of the best-known host defense functions of the complement system is its ability to lyse many bacteria, envelope viruses, and nucleated cells. Lysis of cells by the complement system is mediated by a large macromolecular structure called the membrane attack complex, or MAC. The MAC is formed through what is called the terminal complement pathway, which starts with the generation of C5b. C5b associates with C6, C7, and C8, forming a large multimolecular complex that associates with and begins to disrupt the cell membrane. Subsequently, C9 binds to C8, and this is followed by the binding of many additional C9 molecules, leading to the formation of a large pore in the cell membrane. The pore in the membrane is large enough to allow water, ions, and small molecules to enter the cell, leading to lysis. Activation of the alternative pathway. The alternative pathway of complement is activated by LPS and other lipopolysaccharides found on the surface of invading pathogens. The alternative pathway is always active at a very low level. This activation is due to the spontaneous hydrolysis of an internal thiol ester bond in C3. Hydrolyzed C3, called C3 water, has a binding site for factor B, another protein in the alternative pathway. Factor B binds to C3 water and becomes a substrate for cleavage by factor D, denoted here as FD. Factor D cleaves factor B into two fragments, BA and BB. The enzymatically active BB fragment remains associated with C3 water, and this bimolecular complex is the alternative pathway C3 initiation convertase. Alternative pathway C3 initiation convertase. This initiation convertase cleaves C3 into two fragments, C3a and C3b. The newly generated C3b can covalently attach to nearby surfaces, just as C4b does in the classical pathway. In the absence of an appropriate activator for the alternative pathway, complement regulatory proteins block further activation of the pathway. However, C3b, on interacting with an activating surface, such as a bacterial cell, will bind factor B, which will be cleaved by factor D, resulting in the formation of the alternative pathway C3 convertase of C3b-BB. Alternative pathway C3 convertase. The C3 convertase of the alternative pathway cleaves C3 to C3a and C3b. Some of the C3b generated by this cleavage does not interact with the activating surface and remains soluble. Some of the C3b generated will covalently interact with the activating surface and start the formation of additional alternative pathway convertases, thus rapidly amplifying activation of complement through this pathway. The surface of an activator, such as a bacterial cell, can rapidly be covered with many thousands of C3b molecules in a few minutes as a result of this amplifying effect. And, as with the classical pathway, some of the C3b generated will interact with the C3 convertase itself, resulting in the formation of a trimolecular complex of two C3b molecules and BB. This multimolecular complex is called the C5 convertase of the alternative pathway and is specific for cleaving C5. The C3a and C5a fragments generated by activation of the alternative pathway mediate the same functions of chemoattracting and activating granulocytes and phagocytic cells. C3a attracts mast cells to sites of complement activation, and binding of C3a and C5a to these cells, as well as basophils, induces degranulation and release of histamine and other vasoactive amines. C5a is a chemoattractant for macrophages and neutrophils, and binding of C5a to these cells primes them for mediating their host defense functions. Activation of the alternative pathway results in the formation of the membrane attack complex, leading to the lysis of the invading pathogen. Lysis of cells by the complement system is mediated by a large macromolecular structure called the membrane attack complex, or MAC. The MAC is formed through what is called the terminal complement pathway, which starts with the generation of C5b. C5b associates with C6, C7, and C8, forming a large multimolecular complex that associates with and begins to disrupt the cell membrane. Subsequently, C9 binds to C8, and this is followed by the binding of many additional C9 molecules, leading to the formation of a large pore in the cell membrane. The pore in the membrane is large enough to allow water, ions, and small molecules to enter the cell, leading to lysis.
right? So that was the brief overview of complement activation and what are the major pathways. So we discussed only about the classical and the alternative. There is lectin based as well, so we're gonna cover this. We are going to come back to this particular link after covering the theory. Okay, so let me start with it. So the pathways of complement activation. There are three pathways of complement activation. The alternative and lectin pathways are initiated by microbes in the absence of antibody. And the classical pathway is initiated by certain isotypes of antibodies attached to antigen. Several proteins in each pathway interact in a precise sequence. The most abundant complement protein in the plasma, that is the C3, plays a central role in all three pathways. The early steps of all three pathways function is to generate a large number of functionally active fragments of C3 bound to the microbe or cell where the complement pathway was initiated. By convention, the smaller proteolytic fragment of any complement protein is given the A suffix. So C3 will be cleaved into C3A, which will be the smaller subunit, and the larger piece is the B fragment. So C3B will be the larger segment. Except C2, the alternative pathway of complement activation is triggered by spontaneous hydrolysis of C3 in plasma at a low level. The breakdown product of C3 are unstable and, in the absence of infection, are rapidly degraded and lost. However, when a breakdown product of C3 hydrolysis, called C3b, is deposited on the surface of a microbe, it forms stable covalent bonds with microbial proteins or polysaccharides. The microbe bound C3b binds another protein called factor B, which is then cleaved by a plasma protease called factor D to generate the BB fragment. This fragment remains attached to C3b, and the complex C3b-BB complex functions as a proteolytic enzyme called the alternative pathway C3 convertase. So remember how C3 convertase is being made? It is being initiated by the C3 itself and the factors D and B. Okay. The C3 convertase is stabilized by propyridine, a positive regulator of the complement system. As a result of the enzymatic activity of the C3 convertase, so what exactly is C3 convertase? It is C3b-BB complex of proteins. Okay. Many more C3b and C3b-BB molecules are produced and become attached to the microbe. Some of the C3b-BB molecules bind an additional C3b molecule, and the resulting C3b-BB3b complexes function as a C5 convertases to cleave the complement protein C5 and initiate the later steps or the late steps of complement activation. While in the classical pathway, the activation is triggered by IgM. So remember, there are total 3 complement pathways, 2 of which do not require any antibody antigen complex, while one pathway needs the recognition of antigen antibody complex, and that pathway is classical pathway. Okay. So the activation is triggered when IgM of certain subclasses of IgG, Ig1 and Ig3 binds to antigen. As a result of this binding, adjacent Fc region of the antibodies become accessible to and bind the C1 complement protein, which is made up of binding component called C1q and 2 proteases called C1r and C1s. The attached C1 becomes enzymatically active, resulting in the sequential cleavage of 2 proteins, C4 and C2. One of the C4 fragments that is generated, C4b, becomes covalently attached to the antibody or the microbial surface where the antibody is bound, and then binds C2, which is cleaved by the active C1 to yield the C4b2A complex. This complex is the classical pathway C3 convertase. So till now we have seen two C3 convertase. One is C3bBB. This also acts as C3 convertase, and C4b2A. This is also a C3 convertase, which functions to break down C3, and the C3b that is generated again becomes attached to the microbe. Some of the C3b binds to the C4a2b complex, resulting in C4-2a3b, just like C3b-BB3b complex function as a C5 convertase, which cleaves the C5 complement. Okay, so what you guys have to remember in the complement system is how the C3 convertase is made, like what was the initiation factor, what are the complex components in the C3 convertase, and what are the components of the complex in C5 convertase. Okay, now the lectin pathway. So the lectin pathway of complement activation is initiated not by antibodies but by the binding of circulating lectins such as plasma mannose binding lectin or ficolins to microbial polysaccharides. Serine proteases structurally related to C1s of the classical pathway are associated with these lectins and serve to activate C4. The subsequent steps are the same as in the classical pathway. Okay, so it's just the initiation and the components, they are different in the classical and the lectin pathway. Okay, so before going to this, we're going to see one more detailed representation of the complement system. It will help you in creating a storyline of complement system in your head.
Are you someone who gets anxious every time you need to speak up at
about the science infection into new. The nomenclature for the complement system can be a bit complicated, even for immunologists, so let's break it down a bit. There are 9 main complement proteins, which are named with the capital letter C and then a number. The tricky part is that the number comes from the order in which the proteins were discovered, not the order they appear in the complement cascade, so keep this in mind moving forward. When a complement protein is cleaved, the cleavage products are given a lowercase a or b after the number. The A fragment is always the smaller fragment, an anaphylatoxin, while the B fragment is the larger binding portion, except in the case of C2. For example, when C3 is cleaved, it is cleaved into C3A and C3B, with C3A being the smaller product. As you will soon see, many of these cleavage fragments complex together to form enzymes or other functional proteins. To designate which complement proteins make up a complex, the names of the proteins are strung along in the order in which they bind. For example, the classical C3 convertase is called C4b2A because it is made up of C4b and C2A. When C3b binds to this complex to create a C5 convertase, the complex becomes C4b2A3b. As a quick note, some sources have recently begun swapping the designation for C2 so that the C2A and C2B fragments match the rest of the system, which would make the C3 convertase C4b2B instead. But this convention, as logical as it may be, does not seem to have been officially adopted, so it may be important to be aware of this inconsistency within the field, depending on your specific purposes in learning immunology. Now that we understand the system of nomenclature, let's learn about how the complement system actually works. Complement can be activated through one of three distinct pathways. Each of these pathways converges at the step of C3 cleavage, which kicks off the most important effector functions of the complement system. We'll discuss the activation of all three pathways first, and then circle back to what happens after C3 is cleaved. The classical pathway was the first complement pathway to be discovered. The classical pathway begins with a protein complex called C1. C1 is made up of three distinct subunits: C1q, C1r, and C1s. C1r and C1s are proteases, which are enzymes that cleave proteins, but they are inactive during normal conditions. During an infection, circulating C1q can bind directly to some bacterial surfaces, to antibodies bound to a pathogen, or to an acute phase protein called C-reactive protein that also binds to bacterial surfaces. C1q binding causes a conformational change, resulting in autocatalytic activation of C1r. Activated C1r then cleaves and activates C1s. Once C1s is activated, it cleaves circulating complement proteins C4 and C2. C4b covalently binds to the pathogen's surface or to pathogen-bound antibodies, and C2a binds to C4b, forming the C4b2a complex. This complex is known as the C3 convertase, and it cleaves circulating C3 into C3a and C3b. Although the classical pathway of complement activation was the first to be discovered, it is likely the youngest evolutionarily. After all, it often relies on antibody recognition of pathogens, and antibodies are only found in jawed vertebrates. Since complement can be found in lower-order organisms, there must be other ways for complement to be activated. One of these pathways is called the lectin pathway. Lectins are proteins that can recognize and bind to very specific carbohydrate groups. While some lectins play homeostatic roles within the host, others act as pattern recognition receptors, detecting arrangements of sugar molecules that are only found on pathogens, such as those found on bacterial cell walls or adorning yeast cell surface proteins. So far, we know of 4 lectins that activate complement after binding to microbial sugars. These include mannose-binding lectin, or MBL, and phycolins 1, 2, and 3. Mannose-binding lectin is made in the liver and circulates in the bloodstream. It is a multimeric protein with multiple binding sites that recognize the sugar groups fucose, mannose, and N-acetylglucosamine. These sugar groups are commonly exposed on microbial polysaccharides, but not on host cells. The phycolins are structurally similar to mannose-binding lectin, except that they bind acetylated sugars instead of mannose groups. Now, here's how lectin binding initiates complement. In the circulation, mannose-binding lectin, or one of the phycolins, complexes with the proteins MASP1 and MASP2, which are MBL-associated serine proteases.
बहुत ज़्यादा चिंता होता है कभी गिर जाए कभी लोग
Proteases are inactive until the lectin binds to the sugars on a pathogen's surface. At this point, a conformational change activates MASP2, allowing it to cleave and activate another molecule of MASP2, which can then cleave the complement proteins C4 and C2. C4b complexes with C2a to form the C3 convertase, and here the pathway converges with the classical pathway. The final pathway of complement activation is known as the alternative pathway because it was discovered after the classical pathway. Although it is called alternative, it is probably much older evolutionarily than the classical pathway. The biggest difference between the alternative pathway and the other two is that it utilizes a distinct C3 convertase. This convertase consists of C3b bound to a cleaved fragment of a plasma protein called factor B. When C3b is bound to the surface of a pathogen, circulating factor B can bind. This allows another plasma protein called factor D to cleave factor B, leaving behind the rather confusingly named C3bBB. This is the alternative pathway C3 convertase. You might be wondering how one of the subunits of this C3 convertase is C3b, since C3b can only be generated by a C3 convertase. Remember that C3b can be generated by the lectin or classical pathways. Additionally, spontaneous hydrolysis in C3 can lead to a short-lived intermediate convertase that can also generate C3b. So what happens after the generation of the C3 convertase? Remember, for the classical and lectin pathways, the C3 convertase consists of C4b2A, and for the alternative pathway, it is C3bBB. C3 is the most abundant complement protein in the serum, even with a meager concentration of 1.2 milligrams per milliliter. And because of this, C3 has incredible capacity to amplify early complement events. Additionally, C3b can create more of itself by utilizing the alternative pathway. Remember that both kinds of C3 convertases are bound to the pathogen surface. When it is exposed to circulating C3, the convertase cleaves the C3 protein into two parts, C3a, the smaller part, and C3b, the larger. C3a is released into the circulation, where it recruits neutrophils and other phagocytes to the site of infection. C3b, on the other hand, can do one of two things. The first thing is that many molecules of C3b can bind to the surface of the pathogen, coating it in a process called opsonization. While opsonization doesn't directly destroy the pathogen, it marks it for destruction by phagocytes like macrophages. Phagocytes have receptors that recognize C3b coating the surface of a pathogen and will phagocytose objects or organisms coated in C3b. Opsonization is especially effective when the pathogen is also coated in antigen-specific antibody, as phagocytes also have receptors that recognize bound antibody. The second thing C3b can do is bind to the existing C3 convertase, creating a C5 convertase. As you might imagine, once the C5 convertase is formed, it can cleave C5. Like C3a, C5a is released into the circulation to recruit phagocytes. Meanwhile, C5b nucleates the membrane attack complex, a complex of complement proteins that form pores in pathogen cell membranes, causing the pathogen to lyse. Here's how this works. C5b binds to circulating C6 to form C5b6. C5b6 then binds to C7. Binding causes a conformational change in C7, which exposes hydrophobic residues, allowing C5b67 to insert into the pathogen's cell membrane. C8 then binds the complex, which also causes a conformational change.
It was our early meeting end session, so just give me 1 minute.
Change, allowing C8 to insert into the pathogen's plasma membrane. Finally, 10 to 16 molecules of the terminal complement protein C9 polymerize within the cell membrane to form a pore that allows water and solutes to flow out of the pathogen, leading to death. This is possible because while the outer surface of the pore is hydrophobic, allowing it to insert into the plasma membrane, the inside surface is hydrophilic, allowing polar molecules like water and ions to travel freely through it. Although the membrane attack complex is able to kill many kinds of bacteria and parasites, it is especially important for controlling bacterial infection with Neisseria species and may actually be dispensable for controlling other infections. The evidence for this is that genetic defects in the terminal complement proteins C5 to C9 do not result in increased susceptibility to any known pathogens except Neisseria.
AI isn't.
Oh, we have a question. Let me read it. How does IgE mediated mast cell activation contribute to allergic responses, and what mechanisms are involved in the pathological effects seen in conditions like asthma or anaphylaxis? How does IgE mediated mast cell activation contribute? Okay, it's the same question. Yeah. So we have not yet covered this, like how each and every antibody is doing its function. So what IgE majorly does it, that just like other cells, let me have a reference. So imagine if this IgE and this is a mast cell. Okay, so what it will do, it will recognize the mast cell, the IgE antibody will bind to it, and it will lead the changes, hence the signaling inside the mast cell to release the histamines basically. Okay, so I don't remember exactly whether in the class this topic was covered or not, but I don't think so it was covered, because otherwise I would have definitely put it. So what I can do is maybe in the next class on next Sunday we can have a dedicated one slide of IgE specific responses, because they are majorly two ways through which IgE acts. One, by releasing the histamines, okay, from the mast cell. Second, IgE also helps in controlling the helminths or parasitic microbes, okay, because parasites are quite big in comparison to the bacteria or the virus or other microbes which we have talked about. So you need a specific antibody, which in this case is IgE, which can have function against the helminths, as well as it is able to bind to the mast cell and let the mast cell secrete. So there are granules in the, so imagine, I don't have any representation of it here, but imagine if this is a mast cell, so imagine if this is a mast cell, there'll be, so you can see these vacuoles are here, right? So in the mast cell, these vacuoles will be filled with histamine, okay, and once the IgE antibody comes and bind to the mast cell, it will lead to the release of these vacuoles, and hence the histamine will come into the circulation or the place of your allergic reaction. And histamines are vasodilators, okay, so they will, again, vasodilator means it is, now we are connecting the concept of cytokines, you remember. So in cytokines you need to expand the cells which are lining the blood vessels, so that lymphocytes can come into the place of infection, right, at the place of infection. So histamines, what they'll do is they are vasodilators, so they'll dilate the blood vessels at the localized area, which will lead to the more blood circulation and more heat, okay, and then again it will connect it with the inflammation and with other cells migrating to this place and leading to the handling of the infection or the component which has just caused you the allergy. Okay, I understand this is not the like theoretical explanation of it. I don't have it here with me, but this is the basic idea. I hope whoever has asked that question. Got a little bit of satisfaction with this, Janaki. Okay. But
Yeah, ma'am. Thank you, ma'am.
okay, but if I remember it, I'll keep this IgE mediated response in the next class. Okay. So for now, as you can see, you might feel very overwhelmed with the complement system. It might seems very complex and all, but trust me, it's not. So once you can take the common points from all these three pathways, then it becomes easy. Okay, because at the end of the day, there is formation of C3 convertase. That is a major event. How C3 convertase is being made, it is different in all three pathways. Okay, next step is C5 convertase. So C5 convertase making is same. If you remember the nomenclature, is just the complex will be different. So what you have to remember in this is how the pathway is being initiated, how C3 convertase is made, rest every other thing is same. Okay, so if we look at the alternative pathway, so how the alternative pathway is being initiated, so there will be C3 molecules in the circulation. They'll go and they'll randomly cleave and they'll go and bind to the surface of the microbe. Once they are bound there, then a BB complex, a protein, it will also come and will bind to the C3b. Once C3b and BB complex is there, it will again attract another C3b complex, and now the C3b BB C3b complex is a C3 convertase. Okay, once this convertase is made, what it will do is it will convert the C5. Once C5 has been converted to C5a and C5b, then this will go on and do its work. Okay, it can go and bind to the microbe and do all other, like making the membrane attack complex and attacking other cells which are nearby. Okay, so this was the alternative pathway. And here you can see there is no need of microbe or antibody. There is no need of antibody basically. The complex is not needed, it's just the microbe itself. When we talk about classical pathway, in classical pathway we need the microbe as well as the antibody associated with it. Okay, and once this is present, it is present along with the C1q, 4 and 2 molecules. Okay, now it will make C4b and 2a, they are the C3 convertase in this pathway. The pathway in which the antigen antibody complex is required. So this is your microbe, this is your epitope which your antibody is making. This antibody's one end is associated with the C1q protein, and C1q is associated with C4 and C2, which further makes them into C4b and C2a. Now C4b C2a can cleave the C3 just like C3b and BB complex. Here C4b 2a complex can cleave the C3, making the C3b unit come with the C4b C2a complex. Now C4b 2a and C3b, all these protein complexes together make the C5 convertase, and the same thing will happen. So the only difference was the initiation of it. Whole pathway is same, it's just how it is being initiated is different. Here if we talk about lectin pathway, so instead of the antibody and microbe identification, the proteins are recognizing the mannose or the lectins basically. Okay, so they are recognizing that and then making the, so you can see here the C3 convertase is same in classical pathway and the lectin pathway, is just here the complex is being recognized and here the lectins are being recognized, and the whole pathway is same. Okay, so I hope that the video sessions actually helped you in summarizing these events in a little bit more concise way. Okay. So this is just the basic theoretical functions of whatever we have seen till now. So C3, what it will do, C3b binds to the surface of a microbe where it functions as an opsonin. Now you know what opsonization or opsonin is, and and as a component of C3 and C5 convertases. C3a stimulates inflammation. Factor B. So BB is a serine proteases and the active enzyme of C3 and C5 convertases. So that BB unit is there, right? So it must be coming from a protein. That protein is called factor B. Then factor D. Plasma serine proteases that cleaves factor B when it is bound to C3b. Propyridine. So propyridine stabilizes C3 convertases, C3b and BB on microbial surface. Don't get overwhelmed again, just go to the lectures, sit through them, take one lecture at a time. So they are like 35 to 40 minutes long, okay? So just take one lecture where the complement system is being summarized. So these things will become more clear to you. And if you do not understand or still have little bit of confusion, you can always come to the next class and then we can try to make it little bit more simple, okay? When we talk about classical and lectin, so you know now that classical and lectin, both the pathways are same, even the C3 convertase is same, it's just how they are being initiated is different, okay? So C1, which is associated in classical pathway, so especially the C1q-R2S2, so it initiates the classical pathway where C1q binds to Fc portion of the antibody. C1R and C1S are proteases that lead to C4 and C2 activation. While C4b covalently binds to surface of microbes or cells where antibody is bound and complement is activated. C4b binds to C2 for cleavage by C1s. C4a stimulates inflammation. C2A is a serine protease functioning as an active enzyme for C3 and C5 convertase, while MBL or mannose binding lectin initiates the lectin pathway. MBL binds to terminal mannose residues of microbial carbohydrates. MBL associated proteases activate C4 and C2 as C1R and C1S while they do not play any role in the classical pathway. Saying again, this is there just for your information. The first thing first which you should have in your understanding and also to solve the questions, you should know what are the major components which are initiating the pathway. And by now you have the answer to why this is called a complement system, because it is complementing the already existing humoral immunity, right? So in the humoral immunity we studied all about antibody, how antibodies are being made, what are the different types of antibodies, where they are made, what happens when they are on the cell surface, how exactly the cell decides which antibody to be formed, what functions they are doing, whether it's opsonization, neutralization, complement activation. So now you know that only the classical pathway is the one, right, in which antibody is actually required for its initiation. So the function of antibody, which we saw in the earlier discussion of today's class, there only one pathway is dependent upon the antibody. No other pathway is dependent on the antibody. And it doesn't mean that when complement system is active, the humoral immunity or the antibody associated immunity is not taking place. Remember, the ultimate goal or the ultimate aim is to clear the infection as soon as possible. So while the B cell and T cell, this function is taking place, right, the complement system is also active. Complement system is also working simultaneously. And remember, we talked about the T cells as well, cytotoxic T cells. They are also working. So now at this stage of your immunology course, you can associate different factors. So you remember we discussed about the inflammation, how the inflammation happens. So there you can now add another concept of antibody which might play a role in the inflammation, right? You can now put complement system in the circulation, that a microbe is being subjected in the circulation, so complement system might be taking place, or it might be there. Okay, so don't get confused or don't get overwhelmed that if this is happening then what will happen to that. No, you have already understood different chunks of the major immune responses which as a host we have. Okay, now it's a little bit too much info, but I really want to finish this. What we'll do is we'll summarize complement system in the next class. So take your time, read, take the lecture if you have not yet completed your lectures, do it, and then we'll discuss all this in the next class. Okay, so for today we can jump to our questions. You know the drill. So the drill is basically, I don't remember. Did we complete our questions? I think we completed. We completed our questions, right? Okay. So the normal drill is that for easier questions we'll have 1 minute, for a little bit bigger or difficult question we'll have 2 minutes. I will encourage every one of you to please participate in the quiz. Even if the answer is wrong, it's okay, we always end up having the right answer. So yeah, so first question for today is, multipotent progenitor cells via their FLT3 receptor tyrosine kinase bind to blank cells: stromal cells, mast cells, dendritic cells, or macrophages. So I'll give you 1 minute, because if you have attended and if you are up to date with the lectures, you'll get this answer like in just by looking at the option itself. But
Option A, ma'am.
yes, but I want everyone to play a role in this, so please put your answers in the chat. Okay, so for this question obviously the answer is stromal cells. So this is not exactly how the cells are being generated or proliferated or developed, but I want you to have an idea about the receptor concept. So as you can see, this is a leukemic cell, basically a cancer cell. This is your stroma or the bone marrow. Okay, so even the FLT3 receptor is present on cells, and it can have different functions independent of the function through which we have studied it. Okay, so you can see there are different types of receptors which are present on every single cell. It depends upon the ligand, it depends upon their local, like in which they are differentiated. So every function depends upon the condition. So the same receptor might play one role in different condition, and the same receptor will play different function in different condition. Okay, so moving to the next question. Why I feel that we have discussed these questions? Have we discussed these? No, we have not. Okay, yeah, but you have seen the answer. So I'll directly, so basically the question is, which chemokine helps to retain stem cells and lymphoid progenitor cells at appropriate stromal cells in the bone marrow? Is it CCL12, CXCL12, CXCL3, or CCL2? So the right answer is CXCL12. This particular visual aid is not exactly relevant to our current topic, like the question which we have, but it is for sure needed for our overall understanding. Okay, so just give me 2 minutes.
protein molecules that play an essential role in immune responses and inflammation. They are produced by various cells, including leukocytes, endothelial cells, and fibroblasts, in response to various stimuli such as microbial infection, tissue damage, and inflammation. Chemokines act as chemoattractants, which means they can attract and guide immune cells to the site of infection or injury. They do this by binding to specific receptors on the surface of immune cells and triggering a cascade of intracellular signaling pathways, ultimately leading to the migration of the cells towards the source of the chemokine. In addition to their role in immune responses and inflammation, chemokines are also involved in many other physiological processes, including embryonic development, angiogenesis, and cancer metastasis. There are many different types of chemokines, classified based on the arrangement of cysteine residues in their amino acid sequence. Some of the most well-known chemokine families include CXC chemokines, CC chemokines, and CX3C chemokines. Types and functions of chemokines. 1. CXC chemokines. They are involved in a variety of functions, including inflammation, angiogenesis, and hematopoiesis. CXC chemokines are involved in attracting neutrophils, lymphocytes, and dendritic cells. For example, the CXC chemokine CXCL8 is involved in the recruitment of neutrophils to sites of infection and inflammation. Source: PMID 20553655. Other CXC chemokines, such as CXCL9 and CXCL10, are involved in the recruitment of T cells to sites of inflammation. Source: PMID 241074003. Some subtypes of CXC chemokines include CXCL1, CXCL2, CXCL3, CXCL8, CXCL9, CXCL10, and CXCL12. 1. CC chemokines. They are involved in a variety of functions, including inflammation, immune cell migration, and angiogenesis. CC chemokines are involved in attracting monocytes, macrophages, and eosinophils. For example, the CC chemokine CCL2 is involved in the recruitment of monocytes and macrophages to sites of inflammation. Other CC chemokines, such as CCL5, are involved in the recruitment of eosinophils to sites of inflammation. Source: PMID 270966103. Some subtypes of CC chemokines include CCL2, CCL3, CCL4, CCL5, CCL11, and CCL17. 1. CX3C chemokines. They are involved in a variety of functions, including immune cell migration, inflammation, and angiogenesis. CX3C chemokines are involved in attracting natural killer cells and T cells. For example, the CX3C chemokine CX3CL1 is involved in the recruitment of natural killer cells and T cells to sites of inflammation. The only known subtype of CX3C chemokine is CX3CL1. 1. XC chemokines. They are involved in a variety of functions, including immunoregulation and inflammation. XC chemokines are involved in regulating the immune response. For example, the XC chemokine CXCL14 is involved in regulating the immune response by inhibiting the recruitment of T cells to sites of inflammation. Some subtypes of XC chemokines include CXCL14 and CXCL16. Overall, chemokines play a crucial role in the immune system by attracting immune cells to sites of infection or inflammation. Dysregulation of chemokine signaling has been implicated in a variety of diseases, including cancer, autoimmune disorders, and chronic inflammatory conditions.
So as I've mentioned, it was not directly related to this particular thing, but we may not trickle into the same topic. So I just wanted to take this opportunity and let you guys familiarize with these classes of cytokines. It's not necessary that you should remember it or memorize it. No, it was just a general information. Okay. So the next question for today is: pro B cell mature into pre B cells upon interaction of VLA4 and VCAM1, c-KIT and SCF, both A and B, or FLT3 and FLT3L. And Your 1 minute time starts now. Please put your answers in the chat box.
Can see, ma'am.
We are still going to wait for 30 more seconds, okay? And your time is up. The correct answer is C. Yes. Thank you for participating. So as usual, wherever extra information is needed, we're gonna cover it, and this is another example of it. It helps in revision.
Hi everyone, in this video I would discuss the B cell development. Now, B cell development starts in the bone marrow. Bone marrow is the primary lymphoid organ where immune cells develop. In the early stage of B cell development, it takes place in the bone marrow. Then later, when B cells mature a little bit, it moves out to the secondary lymphoid organs such as lymph node and develops there. So the primary place of B cell development is the bone marrow. Inside the bone marrow, there is endosteal niche. Endosteal niche is closer to the bones, where the osteocytes are there, and there is another niche known as vascular niche around the blood vessels. In the endosteal niche, there are certain cells known as stromatal cells. Now, these stromatal cells play a crucial role in B cell development. So let's just take a moment to learn more about the stromatal cells. The stromatal cells are actually supportive cells, and they are large adherent cells. The Greek word stroma means matters. The stromatal cells work like a substratum, which holds the pro-B cells in a specific niche and also influence their development by secreting several cytokines, which are very important in B cell development. We will talk about these cytokines and the signaling downstream to these cytokines and how they are involved in B cell development. So stay tuned. Now, inside the endosteal niche, there are hematopoietic pluripotent stem cells. And hematopoietic pluripotent stem cells can give rise to lymphoid progenitor cells, which is not shown here. Ultimately, this lymphoid progenitor would give rise to a pre-pro B cell, which is a total immature stage of a B cell. At this stage, B cell expresses some adhesion molecules. Also, this pre-pro B cell expresses a B cell lineage marker B220. Now, also at this stage, B cell expresses another transcription factor which is known as EBF1, and we would learn about EBF1's function and its role in B cell development. At this point of time, the bone marrow stromatal cell expresses adhesion molecules known as VCAM1. On the other hand side, the pre-pro B cell expresses adhesion molecule known as VLA4. This VLA4 and VCAM interaction is very important for B cell development. Now, VLA4 VCAM1 interaction hold the pre-pro B cell near to this bone marrow stromatal cell niche, such that stromatal cell can influence its development. And also, this VLA4 and VCAM1 interaction give rise to expression of another adhesion molecule known as c-KIT, and c-KIT has defined role. From the pre-pro B cell, after the interaction of VCAM1 and VLA4, it starts expressing c-KIT. On the other hand side, stromatal cell expresses stem cell factor or SCF. This SCF and c-KIT interaction is important clue for the proliferative signal to B cell. Also, another chemokine known as CXCL12 mediated signaling is very important for pre-pro B cell. Now at this point of time, two important phenomena happens. One is c-KIT and SCF interaction, which give rise to a proliferative signal, and also at this point, the CXCL2 based signaling system give rise to proliferative signal. From this point onwards, the heavy chain DJ recombination starts. Now as time progresses, the early pro B cell detaches from that specific stromatal cell and goes and bind to other stromatal cells which are IL-7 secreting. Now IL-7 has distinct role in early pro B cell development. IL-7 receptor is situated in the early pro B cell, and IL-7 binds to IL-7 receptor, and this gives rise to specific signaling. At this point of time, VDJ recombination takes place. So the B cell receptor, which is basically membrane bound anti-IgM or anti-IgD, they are actually produced, the heavy chain is produced from this point onwards. So let's just take a look what IL-7 mediated signal do. So IL-7 bind to its specific IL-7 receptor on the early pro B cell stage. Now it binds to the IL-7 receptor, which is a tyrosine kinase, and which is a tyrosine kinase based receptor. Now along with it, Janus kinase is associated. Now JAK gets phosphorylated upon IL-7 binding, and it allows phosphorylation of STAT5. STAT5 moves to the nucleus and start transcription of NMYC and CMYC. Also, it start transcribing EBF1, and EBF1 and STAT5 both allow transcription of PAX5. EBF1 is super important for DGH recombination, and also PAX5 is very important for VDJ recombination. For the heavy chain recombination, PAX5 and EBF1 are super important. So these two transcription factors ensures that the heavy chain recombination takes place properly. Now EBF1, E2A and PAX5, all these transcription factors actually activates many genes which are important for B cell lineage specification and B cell proliferation. Now, once the early pro B cell stage is over, then comes the late pro B cell stage. In the late pro B cell stage, it starts expressing a primitive version of BCR, which is known as B cell receptor. Now, at this point of time, you see the heavy chain of the B cell receptor is formed, but the light chain at this point is not properly formed. So at this point,
So all this information you already have. We have very well discussed. Okay, why we are repeating it multiple times? So that we have a memory of things and we can easily go back to the sections where we have studied them or read about them and recollect our concepts. Okay, so this is it for today, I guess. Okay, so it was a lot of information, I understand, but please be up to your lectures, and yeah, that's it from my side. Thank you.